I wrote this book to try to give a flavor of the field and give people an idea of what is going on inside an embryo's brain. Because every baby is born with its own brain. Like you're born with a particular face that might be a combination of your father's, your mother's, but it's partly your own too. Your brain is also your own. Because of all the complications, the thousands and thousands of steps and molecules and genes involved, no two people's brain is ever going to be built the same way. So it's just interesting for people to understand that we're not born just as pure, blank, equal slates. Everybody's different in their own way.
Welcome to Brain Science, the podcast that explores how recent discoveries in neuroscience are unraveling the mystery of how our brain makes us human. I'm your host, Dr. Ginger Campbell, and this is episode 201. Today's episode is an interview with William A. Harris, who is a professor emeritus from Cambridge University. He will be talking about his new book, Zero to Birth: How the Human Brain is Built. You can send me feedback at brainsciencepodcast@gmail.com, and please visit my website at brainsciencepodcast.com for complete show notes and episode transcripts. You can get Brain Science show notes automatically every month if you sign up for the free Brain Science newsletter. You can do this at brainsciencepodcast.com or just text Brain Science, all one word, to 55444. When you sign up, you'll get a free gift entitled 5 Things You Need to Know About Your Brain. That's Brain Science, all one word, to 55444. Because Brain Science is independently produced, it relies on the support of listeners like you. You can learn more at brainsciencepodcast.com/donations. I'll be back after the interview with a summary of the key ideas and a few brief announcements. Bill Harris, it's great to have you on Brain Science today.
Thank you very much, Ginger. It's great to be here. Thank you for having me.
So before we talk about your new book, Zero to Birth: How the Human Brain is Built, I'd wonder if you would mind just telling us a little bit about yourself.
Myself, I'm a 72-year-old retired professor of anatomy at Cambridge University. I studied in the field called developmental neurobiology throughout my career, half of which was in California at UCSD. And then for the second half, I moved to the University of Cambridge here in England. I've studied, I don't know if you want to know my scientific career or about me as a person more.
Well, tell us about what got you interested in developmental neurobiology.
That was when I was a graduate student. When I was a graduate student, I was working with fruit flies in the laboratory of Seymour Benzer, and I was studying color vision in flies. Can flies see different colors? Can they avoid a shock that's associated with one color? Can they learn colors? Can they see them? I was working with some mutants that were colorblind, basically, colorblind flies. And at that point, I wasn't really interested in development, but there was a gene and there was a colorblind fly, and something happened between the gene and the fly going colorblind. And when I started to do a little investigation, I found that one of the cells that was supposed to be a color-sensitive cell had turned into a different type of cell. So how did it turn into this different type of cell? That was a developmental issue. And that was my first kind of glimpse that when you study mutants in the brain, or mutant animals that behave differently, or it's not only identifying the gene, but it's identifying what the gene does, and does it have a developmental effect that causes something to go wrong. So that's how I started to get interested in this whole issue.
I think like the second or third episode of this show, way back in early 2007, I did a brief review of a book that talked a little bit about some of the brain development. And this is the first time I've had a chance to to see the new stuff, since I realized that the stuff that is in your book is not really new, but it's new enough for people like me who are not in the field. So I'm excited to just go through what we've learned. So could you just give us an overview of your book?
It's basically you start at a single cell, a fertilized egg, and you end up with a human brain. And in that process, between going from a single cell to a fully formed human brain, and when I say fully formed, I mean almost fully formed, because we're going to a neonatal brain, basically, at the time of birth. At that point, a lot of the work is done. There's still a fair bit to do, but most of it is done. So what are the steps? How would you build this most complicated machine that we know of yet in the universe? How would you ever build such a thing? That's really the question that motivated me. And developmental neurobiologists over the years and evolutionary biologists and geneticists have found that you can put it into stages. Like in a builder's manual, you might say stage 1, put the legs on the table, and stage 2, do something else with the table. So it's broken into these stages. We call them stages. They really flow into each other. But the book is organized into successive stages, starting with the egg. The egg then makes lots of cells. Some of them become the primordium of the nervous system. Those cells that are the primordial cells of the nervous system then proliferate until they make enough cells to make a full brain. And as they're proliferating and making cells, they're also turning into neurons and glia, the two main components of the brain. And there's thousands and thousands of different types of neurons and glial cells. So we go into the question of how does a cell know to become this type of cell or that type of cell, a rod or a cone in our eye, for example. And after that, then the cells are born and they kind of know who they are, and then they have to wire up. The brain has to wire up. So what is that process all about? How do nerve cells find and make connections with each other? And from there, we go on to more about fine-tuning, how those connections are fine-tuned through experience and activity processes that are happening before there's experience. And that kind of summarizes it. And then the last chapter is specifically about humans. Most of the early stuff is about general principles of brain development as they apply to humans. But the last one is what makes human brains, how do human brains develop differently than other animal brains? So that's the book in a nutshell.
Okay, great. So that's a great overview. So before we talk about how the human brain develops, would you tell us a little bit about the relationship between our brains and single cell life forms? Because there is an evolutionary continuity, right?
Yes, and the book is full of little evolutionary stories too. This is one that sparked my imagination. There was a scientist who worked on paramecium. Xing Kung was his name. He showed that paramecia, these single-celled organisms, who are relatives of organisms that lived in the era of life when there were just protozoans on Earth, already had action potentials. That is, the electrical impulses that neurons have. They use calcium ions instead of sodium ions to generate their action potentials, but they have voltage-sensitive channels in their membranes so that when they get excited a little bit, they have an explosive impulse that travels all the surface of their membrane. This helps these little organisms navigate in their world and make decisions about where to go next and things like that. And it turns out that a lot of components, from the impulse activity that neurons have to the spitting out of neurotransmitters by which one neuron talks to another, evolved in unicellular animals long before there were nervous systems. And it is those genes, or relatives of those genes, descendants of those genes, that work the magic in our brain.
Do you know Seth Grant?
Yes.
I think he might have the record for the most often guest on this show.
Really?
I think he's been on 5 times. And he's very unusual in that he's like one of the very few basic scientists I've ever interviewed that has never written a book. Most of my guests have written books. I'm hoping someday he will write one since he's such a great teacher. But he was the one that first introduced me to this idea that the neurotransmitters and all these structures in the synapse, a lot of them are ancient.
Exactly. And he traced synapse development through evolutionary time by looking at organisms on the tree of life and seeing which components of the synapse had evolved in that species already.
And so then by the time that we get to the vertebrates, we have a nervous system where there are certain structures that all vertebrate nervous systems have, correct?
Correct.
We'll come back to that in a minute. It's just an idea that I want to emphasize as we go along.
And even before vertebrates, the flies, you would say they have a protocerebrum, a deuterocerebrum, and a tritocerebrum. First, second, third cerebrum brain, basically. And we have a forebrain, midbrain, and hindbrain. And actually, the ancestors of flies and vertebrates had a structure already like that.
So let's talk a little bit about what happens at the very beginning. I've read these words as a medical student, and it's never really been very meaningful to me. But would you just take us through the first few weeks of what happens when the fertilized egg is getting the very beginnings of the nervous system?
In a human embryo, there's a group of called the blastocyst. And the blastocyst are basically the stem cells of the embryo. And those cells, we call them totipotent cells because they can make all the different structures of the embryo. Or you could call them pluripotent cells because they can make so many different types of tissues. They can make liver, bone, blood, guts, brain, skin, etc. At some point in development, those totipotent cells, those cells that are able to make everything, some of them become committed to make just one tissue. They start to become specialized in what they'll make. So there's the progenitors of the bone stem cells, or the progenitors of the blood stem cells, the hemopoietic lineage, or the brain stem cells. So that happens for the brain at about the stage of development called gastrulation. That's a stage in development where this ball of cells, which was a blastocyst, some of them dive inside the ball and become internal tissue. Imagine you have a balloon in your hand, a soft balloon, and you push into the balloon with some fingers. So you're pushing some of the balloon inside, right? The part that you're pushing inside is going to become muscle and a tissue that's called the notochord, not nervous system. But that tissue has a role in talking to the cells that it's next to, and it tells those cells to become neural tissue. At that point, it's still a ball. It's kind of like an orange. You could think of it as an orange, and part of the skin of the orange is the neural plate, this flat sheet of tissue, some of which, so if you draw a circle on an orange, this is how I described it in the book, on the peel of an orange, and you think of the cells outside that circle, they're going to become the skin, the epidermis, and the cells inside that circle of orange peel will become the neural tissue. And it starts out flat, like a plate. But then the edges of the plate roll in from each side, the left and the right side, to turn it into a tube-like structure. And that happens at about 4 weeks of gestation in humans.
And this is a stage that all vertebrates go through, but the timing might be different depending on the species.
Exactly, yeah. Some species go through development really fast, some slow.
This month, Brain Science has a new sponsor, Masterclass. With Masterclass, you can learn from the world's best minds anytime, anywhere, and at your own pace. You can explore science with Neil deGrasse Tyson or Bill Nye, or expand into new areas like history, cooking, music, and filmmaking. I've just started exploring Masterclass, and right now I'm deeply immersed in U.S. presidential history and leadership with Doris Kearns Goodwin. One cool thing is that the lessons are short, 10 to 15 minutes, so you can squeeze them into your busy life. But you can also binge on an entire 3-hour class if that works for you. I think you'll be surprised at the diversity with over 150 different instructors teaching everything from dog training to filmmaking. Plus, you can watch them wherever you like, on your phone, tablet, Apple TV, computer, and on the go with the audio mode. I highly recommend you check it out. Get unlimited access to every class, and as a Brain Science listener, you get 15% off an annual membership. Go to masterclass.com/ginger now. That's masterclass.com/ginger for 15% off Masterclass.
Some species go through development really fast, some slow.
So that layer that's going to become the nervous system is called the ectoderm. Most people that have taken biology have learned that. What makes the ectoderm make neural stem cells?
So there's the ectoderm. The ectoderm gives rise to the skin, and it gives rise to the nervous system. It's the epidermal ectoderm and the neural ectoderm. And sometimes in the book, I just say neuroectoderm because that's what we say in the field. I try to explain all these things carefully in the book before I
You do a great job, and I'm just trying to hit the highlights without making things too confusing.
Yeah. Okay. So what makes them become neural ectoderm? They would normally, it seems, like to become skin cells. But there's this factor that's being secreted by the cells that went inside, and it's telling them not to become skin cells. Ignore the signal, which is telling you to become a skin cell, and do the most important thing that it is for you to do if you don't become a skin cell, which is become the nervous system. So these cells are protected from becoming epiderm and allowed to express their default fate, we say, which is to become neural tissue, or the precursors of the brain. So it's neural ectoderm. It's still a sheet of cells or a tube of cells. And these cells are not neurons yet. They're not glial cells. They're just dividing cells, but they're committed now to do this one job to make the brain.
Let me ask you, I think this might be a really stupid question, but one of the things I find most fascinating is the fact that every cell in our body contains the entire genome, right? Which means that if the right genes were turned on, a cell in your body could be something else from what it is. That's pretty wild. When did we figure that out? Was that something that was figured out when they actually got the human genome? How long have we known that?
No, that was long before the human genome, and the key experiment that was done to prove that was the case was first done by a guy named John Gurdon in frogs. And what he did was he let a frog develop, and then he took some skin cells, some deep skin cells, and he took the nucleus of those skin cells and he put it into the egg of another frog whose nucleus, the egg nucleus of which he'd taken out. So he replaced DNA from a random cell in an adult frog, basically, into an egg and got a whole new embryo out of it. It was the first cloning experiment. So he did that with albinos. So he used nuclei from an albino frog and he replaced it into a pigmented mother's egg. And the result was an albino frog. The problem was that his work got eclipsed when they did the similar experiment with Dolly the sheep. And I went to Gurdon's 70th birthday party or something like that. Maybe it was 75th birthday party. And one person said, who was giving a seminar, said, the only mistake that John made was that he didn't name the first frog that he cloned. But he did go on to win the Nobel Prize for that. And then he went with a guy named Yamanaka, who found that you could take a human cell, almost any human cell or a mouse cell or a rat cell or almost any vertebrate cell, expose it to some factors which will reprogram it back to becoming like early totipotent stem cell that can make a whole embryo.
And that's the kind of discoveries that have made the cloning stuff possible.
Indeed.
So I guess we should be getting back to the nervous system, because otherwise we'll eat up. There's just too much fascinating stuff here. Okay, so we're at the stage where we have the notochord and we have neural stem cells. What happens next? Have I got that wrong?
No, well, a little bit, but neural tube is better than notochord. Notochord is underneath. The neural tube, yes, it's full of these neural stem cells and they're dividing. But it's a long tube because an embryo has a head and a tail, a length to it. It's got bulges in this neural tube, big bulges at the front end, which will become the brain, and a long, thin tubular structure at the back end, which will become the spinal cord. So it's getting patterned. There's the brain part, there's the spinal cord part. When you think of an adult nervous system, the neuroanatomists, and I used to teach neuroanatomy when I was teaching, in the human brain, there's hundreds, thousands of parts of a brain, and they're in difficult relationships to each other for students to learn and learn all the names of. Anyways, there's lots of parts of the brain, lots of little different areas. When that's all getting done at this very early stage, it's getting patterned. Like someone was, you take this little tiny structure, which is maybe a few million cells at this point, and start drawing little color patches on it. This part's going to be this part's going to be this part's going to be that. So that's all happening at this very early stage. At the same time, the cells, the neural stem cells that compose the neural tube are dividing and dividing. 2 cells become 4, become 8, 16. Pretty soon there's millions of cells being made every minute until the thing grows into the right size and with all the right regions. So that's discussed in chapters 2 and 3 of the book mostly.
You talk about the fact that there are gradients that sort of tell the cells which end they're at. Could you talk a little bit about that?
The way that developmental biology works seems to be that there are cells that secrete signals and other cells that listen to signals. So cells are talking to each other, deciding you be that, you be this part of the brain, you be that. We have to draw a boundary between one part of the brain and the next part of the brain. And that starts usually with a gradient of a molecule called a morphogen. It's secreted by one place and it's destroyed at another place. So you may have a making enzyme for this signal at one place and a degrading enzyme for this signal at another place. So between these two places, there's at one end, there's a high level of the signal, and at the other end, there's a low level. And then cells pay attention to a threshold. If I see enough of this signal, I'll turn on this gene, which will make me this part of the brain. If I don't see enough of this signal, I won't turn on that gene, so I'll become this other part of the brain. So things are working that in various angles and dimensions to get all these parts of the brain. So that's kind of how it works. More details in the book, of course.
Now, I don't necessarily think that we want to get into a lot of that detail. One of the genes you talk about is the Hox gene, which that's a big one for making sure the head is different from the tail, right?
Yeah, there's a set of Hox genes, first discovered by a guy named Ed Lewis, working at Caltech, and he was working with flies. And he was working with flies that turn one body part into another. So normally flies have 2 wings, but here he'd found a fly that had 4 wings. And here was a fly that had legs coming out of the front of their head. So what was going on? So he found these genes, and they're now called the Hox genes. They were called homeotic genes first, because homeosis is the Greek word from turning something into something else, one part into another. Now, the interesting thing about these genes is they're arranged in a sequence on the chromosome. So there's gene 1, and there's Hox gene 1, and there's Hox gene 2, Hox gene 3, Hox gene 4, Hox gene 5. And each of those Hox genes is dedicated to a specific part of the body. So one of them may be for part of the abdomen, to make sure that that abdomen has abdomen-like qualities. Another one might be in the thorax, to make sure that that part of the fly has thorax-like qualities with a pair of wings, for example. And they go from head to tail. And humans have these Hox genes, the same ones as flies have. We have 4 sets of them. Flies have 1 set of them. And they're arranged in the same way. So the low number Hox genes, like Hox genes 1 or 2, are active anteriorly towards, or rostrally towards the head. And genes with high numbers, like Hox 12 and 13 and 14, are active near the posterior part. The other thing I should explain about Hox genes, if you want me to go a little further, is that they're not morphogens. They're not percolating molecules. They are what is the result of receiving those signals. Hox genes are what are called transcription factors. The genes produce proteins. The proteins then bind to other bits of DNA and turn on other genes. So maybe Hox gene 1 turns on 1,000 genes that may be involved in brain development. And Hox gene 14 or 13 turns on 1,000 genes that are involved in spinal cord development in the posterior part.
The reason I picked the Hox gene is the fact that it also illustrates another important principle that I actually learned from Seth, which is this idea of the gene duplication that happened probably back at the beginning of the vertebrates. Could you talk a little bit about why that's so important?
That's one of a mechanism of, it seems, of evolution. So sometimes a gene will get duplicated. Exactly how that happens in evolution isn't known, but it happens sometimes in the lab when there's sometimes genes recombine, and they mismatch a little bit. So maybe one segment gets paired with another segment, and that ends up causing a duplication maybe in one daughter cell and a deletion in another daughter cell. So part of a gene gets duplicated, or a whole gene gets duplicated. Sometimes a whole chromosome will get duplicated, and because the cell doesn't divide properly, or sometimes the whole set of chromosomes. So some animals are tetraploid. They have 4 sets of each chromosome instead of just 2. And some are even octoploid. They have 8 sets. So these duplication events occur at a large scale or a very small scale. And often gene duplication events, like the Hox genes, we think they started out with probably 1 Hox gene, and then it divided and there were 2, and then those 2 did slightly different things from each other. They evolved slightly different functions. Okay, I'll make this region slightly different than that region, or this cell slightly different from that cell. So they duplicate and then they change, and if that's good and it helps the animal survive, it may then be transmitted to future generations.
So if you have a duplication of a gene, then you can have one of them change and you still got the original that did whatever it needed to do. So this gives you a way to get genetic variation that's more likely to be an animal that would survive.
Yes, it could be. Yeah, definitely. Sometimes these duplicated genes don't do anything, and they're just dead genes that are just hanging around, and they're not doing anything. They're not even red anymore. But sometimes they develop a secondary useful function, not just the original function, but a secondary function that is beneficial in somehow.
I just think it's really interesting that these 2 gene duplications seem to date back to about the time when vertebrate life emerged and became so much more complex than what went before. And there has to be a connection.
Indeed. It was before vertebrates emerged that the Hox genes emerged, as I said. It goes way back to almost the origin of animals.
I want to take a moment to mention Text Expander. Text Expander has been sponsoring Brain Science for over 3 years. Text Expander is a cross-platform app that makes it easy to create short snippets that expand into any sort of content you might want to use. This can range from signatures to full-length documents that include graphics and links. I literally use Text Expander every day, but since I work alone, I tend to forget to mention that Text Expander is also a great tool for teams because you can easily share your snippets not only across your devices, but with other people. If you want to give Text Expander a try, just go to textexpander.com/podcast to learn more and get 20% off your first year. That's textexpander.com/podcast. And don't forget to tell them that you heard about Text Expander on Brain Science. You told us that the Hox gene is a transcription factor. It's not a morphogen. So would you tell us again what a morphogen does? That's the thing that makes the gradient?
Let's give an example of a morphogen. Retinoic acid is a morphogen. It's a metabolic product of vitamin A or carotene that you get from eating carrots. You break it down and then you change it a little bit with different enzymes and you get a product called retinoic acid. And this retinoic acid is a small molecule that can leak out of a cell and diffuse through the cellular environment. So if you're making a lot of retinoic acid at one place and destroying a lot of retinoic acid in another place, then you have a gradient of retinoic acid that's percolating around the cells. The retinoic acid that's at high levels percolates into the cells nearby, and it binds, when it's inside the cell, a molecule of retinoic acid will bind a retinoic acid receptor, which is in the nucleus or goes into the nucleus and turns on the right Hox gene for that concentration of retinoic acid. So it kind of works like that. So the Hox gene then is a gene that produces a protein. It's the protein that is the transcription factor. The gene is the code for the making of that protein, which is the transcription factor.
I guess I misspoke on that one.
No, it's okay. I misspoke too.
Yeah, it gets a little...
It's easy to misspoke and call genes by their function, and you call a gene a transcription factor, but that's not quite right. So I just wanted to be clear about it.
But I'm glad you used the retinoic acid as an example because it just came to me as I remember reading that was the first one of those that you described in the book as the thing that kind of helps the animal develop the head versus the tail. And so I'm thinking that might be why it's not a really good idea to eat lots of vitamin A when you're pregnant. Could sort of screw things up.
They advise pregnant women not to overdo vitamin A, and they also advise them not to get treatment with, what's it called?
Accutane,
Roaccutane, which is a cream that you can put on for acne, and it's very effective treatment against acne, but also because it affects the front versus back development of an embryo. It's also what's known as a teratogen. It causes embryonic malformations. And you put on just a tiny bit of this on a variety of vertebrate lab animals like, embryonic frogs or embryonic fish, and it causes severe deformations like the loss of the head. And when Roaccutane was first developed, a lot of women that were pregnant were probably advised not to use it, but not in the strictest way because they didn't know how bad it was going to be. But then there were a lot of miscarriages and babies with defects. So there's much stronger warning, and it's prescribed.
Could you talk a little bit about what we've learned about how eyes develop and how that's changed ideas about the evolution of the eye?
Yeah. So when I was starting out, I was working on flies' eyes. And although I thought maybe there were some general principles of cell biology that would relate to other animals. I never thought that a fly's eye evolved from the same kind of evolutionary pattern that a vertebrate eye did. Our eye is a camera eye with a lens at the front, which focuses an image on a retina at the back. And a fly's eye has lots of these little facets, and each one has its own little lens and its own set of cells. And they come from different parts of the embryo. A vertebrate eye comes from the front part of the neural plate, and the fly eye comes from a ventral region, what's called a disc inside the fly. So people didn't think there was any chance that they had shared evolutionary history. When I was working on fly's eyes, I started to get in touch with the genes that made the opsin proteins, the photopigments in the fly's eye. It turns out they're the same basic kind of protein that we have. Well, that suggested that maybe opsin genes, the ability to capture light with a protein, and calling them opsin genes, again, they're genes that make a protein that binds a factor called retinal, which is why you have to eat some carrots in order to prime these proteins in your eye so that they can capture light. They probably evolved from an ancient opsin gene. And at the same time, there were mutants in flies that were eyeless. And it turned out that some of them coded for transcription factors, particularly one called PAX6 was famous. It was studied in the lab of Walter Gehring. And this eyeless fly turned out to be related to a mutation in a gene called PAX6. And PAX6 was known in humans to cause eye defects. So all of a sudden, now there's another connection, something deeper than the opsin itself, something that may turn on the opsin that was shared between flies and humans. And you can trace this back, as Walter Gehring did, practically all the way to bacteria when PAX6 first evolved and bacteria first became sensitive to light through bacterial rhodopsins. So amazing. Walter Gehring did experiments like he took the human PAX6 gene and he put it into an eyeless fly, and the eyeless fly could see again. It had eyes again. Or he made the PAX6 gene be expressed, he and his colleagues made the PAX6 gene be expressed in tissues like legs that would normally make legs, but now they made eyes instead of legs. Amazing. And we did similar experiments in our lab too, where we took genes like PAX6 and a few others like that were transcription factors high up in the eye formation pathway and put them into different tissues. And we got ectopic eyes in frogs, like a frog with an eye on its belly instead of in its head, in addition to the ones on its head, I should say.
That's pretty wild, because we used to think that eyes had evolved independently like 40 times or something?
Yeah, that's because there was this huge variety of eyes. You know, squid have eyes, jellyfish have eyes. I was looking at some snail eyes today in our pond. They all have PAX6 genes as part of their mechanism for development. So evolved a long time ago, this gene and several others like it that helped eyes develop.
Yeah, it's really pretty amazing that these basic patterns have been conserved in vertebrates and invertebrates for like hundreds of millions of years. I mean, that's pretty amazing.
Indeed. Single-celled animals before them for almost a billion years, yeah.
So we've gotten to the point where we're at a month in a human embryo, and we've got a neural tube and a body that's got a head-to-toe structure. Now, how are those neural stem cells going to become neurons?
How are they going to become neurons? Okay, so you can imagine a cell that's a neural stem cell. And what this neural stem cell does, as I've tried to explain, is it divides into two, so the brain is growing. But at some point, one of those dividing cells has to stop dividing and say, enough division already, I'm going to become a neuron. I'm going to stop dividing and become a neuron. Once it does that, it never divides again. So you make a neuron in your brain and it stays with you never divides again. And we'll talk about later, it doesn't get replaced either generally. As more and more neural stem cells and their daughters make decisions, I'm not going to divide again, those cells come out of the division cycle, out of the proliferation cycle. They start to become the maturing neurons and cells of our brain, neurons and glial cells of our brain.
So what does the brain look like at birth in terms of these neurons?
Well, actually, when a human baby is born, it has more neurons in its brain than it ever will, because there's not very many more cells being born, not very many new neurons being born. And when I say being born, I mean stopping proliferation.
The division part's already over.
Yeah.
Does that happen before we're born normally or around the same time?
Most of our brain is already born before we're born in terms of neurons. Glia are a different story, but the neurons, which are doing I think they're doing most of the exciting information processing work. Some people that are glial enthusiasts will think glia have a major role in that, but the neurons are the ones that are really electrically active and signaling and synapsing and things like that, and making long-range connections. Anyways, most of those neurons are born. There's few that are born after birth, mostly in the cerebral cortex. The remnants are being born, but there's already a lot that are dying at that point because we generated too many, which we can talk about later. So most of them are born, and all the different regions of the brain are already there. Hundreds of different regions of the brain are already there, and they're each being populated with their own specific subsets of types of neurons. So if you take an example like our retina, the tissue at the back of the eye, the neural tissue at the back of the eye that has the rod cells, which are good for seeing in black and white and low light levels, and then the cone cells, which see red, green, and blue. So those are 4 types of cells that are photoreceptors. Those photoreceptors are neurons that send their signals to about 18 types of next order cells. And then there's about 30 types of the next order cells and another 30. So there's hundreds of cell types in the retina before you even get a connection to the brain. And those are retinal specific types of neurons that have specific jobs to do in the visual system, way at the front of the visual system, at the front end of the visual system. And then you've got thousands in your visual part of your brain that are doing other jobs with respect to processing that visual information.
So how does each neuron know what kind to become?
That's a great question. That's what I spent a long part of my career looking at. I'll tell you a story that I sometimes tell my students, which is, it goes back to my graduate school days. And remember, I was talking about these colorblind flies. Well, one of these flies couldn't see very well in the ultraviolet light. And it turned out it was missing a specific type of cell. Other labs started to pick up on this, particularly Jerry Rubin's lab, and said, well, let's look at that gene. What's that gene doing? And they found what that gene did. It was an enzyme of some sort, a membrane enzyme. And somehow that linked to the formation of this type of cell. But just, it didn't explain it. So they looked for other genes in the pathway. And pretty soon there were like 50 genes, and it took his lab and other labs 10 years to figure out most of the pathway to say, what's important for this cell to become this one type of cell in a Drosophila or a fruit fly retina. And we don't have that kind of information for any cell in the human brain. And imagine how long it would take, I say to my students, to figure it out for each of the thousands of different cell types that we have in our brain. So there's a lot of mystery to it. But again, you can break that process up into stages. First, you decide to become a neuron. Then you decide to become a neuron in the eye. Then you might decide to become, or you could say a motor neuron. Let's do motor neurons. Decide to become a motor neuron because you're in the spinal cord, in a particular region of the spinal cord. Then you have to decide, am I going to innervate a flexor or an extensor? And then you can decide which specific muscle a cell has to decide all these different things. It can be a type, a subtype, a sub-subtype. And those types have generally different morphologies, anatomies. They make different connections, and they drive different information to different parts of the brain.
It's not any one thing that determines it.
No, it's a huge thing. And the things that are important are, we know this, that lineage is important to a certain extent.
But not in Drosophila.
Not in the Drosophila retina, but in lots of other places in the Drosophila nervous system.
This episode of Brain Science is sponsored by BetterHelp Therapy Online. On Brain Science, we often explore how we can keep our brains healthy, such as with exercise and learning new things. But sometimes we can't go it alone. That's where BetterHelp Online Therapy comes in. BetterHelp is convenient, accessible, affordable, and entirely online. All you have to do to be matched with a therapist is fill out a brief survey, and you can switch therapists at any time. Although I haven't used online therapy, I have found working with a therapist valuable in the past, so I feel good about sharing BetterHelp with you. Get unstuck with BetterHelp. Learn more and save 10% off your first month at betterhelp.com/ginger. That's BetterHelp, HELP.com/ginger.
So you have basically a neural stem cell, and this neural stem cell may already be committed to a certain extent to making some general type of cell, but not specifically which one. But it can't make other ones, so if you're derived from that cell, you have limited fate choice. It's like if you were the son of two medical doctors, and they told you, you better become a medical doctor too, and don't do this and don't do that. You might feel that you have limited fate choice. So your ancestors can influence you, and they usually do that by, having a factor in the cell. So when the cell divides, you have that factor too. And then there's environment, because you're getting signals from these other cells, like I talked about morphogens. And the morphogen, if you have two choices, I can be this or that, maybe a morphogen says, okay, become this one or that one. So there's signals. And those signals then turn on other intrinsic things. And over a course of several generations, a cell may be predisposed within a narrow range of fates. And some of those decisions are also based on pure chance. You could be exposed to the same things, but we know that genes are not always, they're not so strict. They go on and off. They flicker on and off. And if they flicker on enough, they may set into motion a little cascade that tells the cell to become one thing rather than another. A great example is the red and green cones in our eyes. When those cells are born, they know they're going to be either red cones or green cones, but they don't know which one. And something just by chance, like the flip of a coin, tells one to become red and one to become green. And Jeremy Nathans discovered the mechanism of that. It's just a little piece of DNA that swings into place, and it can swing this way or that way. And if it swings one way, it's going to be a red cone cell. If it swings another way, it's going to be a green. 50-50, and that's why we have about the same number of each, but you can't predict whether a cell is going to become one or another. And that kind of stochastic or randomized decision-making is happening all over the place in the brain. But because there's so many cells, if you flip 1,000 coins, it's hard to imagine you'd get exactly 500 heads and 500 tails, but you get very nearly there. So even if you have a mechanism that works totally by chance, you can pretty much assure, if you have enough of them, if you do the experiment enough times, that they'll be roughly the right proportions of all the different cell types you need.
So once the neurons get differentiated into the different types of neurons, obviously they've got to now connect to each other. And that's, of course, its own big area. And I haven't done a very good job of time management today.
I'm sorry.
So that's my fault. So could you just give us kind of a overview of how the neurons manage to connect to the right place and not connect, for example, to themselves?
Yes. So the problem of neural connectivity is a huge one and still a huge one. We know that the embryonic brain connects up well. We also know that an adult human being, if you damage the axons, those are the parts, the wires from neurons that go out that send impulses and make connections with other cells. They don't grow well in an adult. So they do something well in an embryo. They get to the right place. They make the right connections. When a nerve cell is born, it's kind of a round cell at the beginning, but it starts to send out these processes, these extensions, one of which becomes the axon generally. And this axon may go for a long distance. At the front of the axon, this extending bit of the cell, there's a machine, a little part of the cell called the growth cone. And it's like an amoeba that's attached to the cell. And it's feeling its way in the environment. It sends out these little antennae called filopodia, and it senses the environment. And it reads where it is. And it makes a decision to grow. Okay, I'm going to grow north for a while. I'm going to go dorsally for a while. Okay, then I'm going to go caudally for a while. I'm going to go west. So it's like a driver that's following signs on a road. It's paying attention to all the signs. So this growth cone is paying attention to all the signs, and it's following. It's also a motor, so it is like a car in a way. It's driving forward, and it's reading the signs where it is, where it wants to go next, until it gets to its home, its target, where it finds its partners and makes synapses.
So there's basically going to be some sort of chemical signal at the target that it recognizes? I mean, is that the basic idea?
There's chemical signals all the way along, all along. So imagine, this is the way I imagine it. I imagine you have a map in front of you, say the map of the city that you live in, and there's all these signs and signals that you can follow if you want to get from place A to place B. And some axons are going from A to B, and some are going from B to A, and some are going from C to D, but they're all using the same map. They're all reading the same map, which is a complicated chemical map of position in the brain. And the factors that are making this map, we tend to call guidance cues, because the axons, the growing axons, are paying attention. Say, don't go here. This is a place to avoid. Come up this way because there's a gradient of this molecule that's pulling you this way. Stick to me because I'm going in the right direction. So there's all these kinds of signals that are guiding the axon, and the axon is paying attention to get to where it wants to go. And that's because it knows who it is as a particular type of cell. I know that I should pay attention to this molecule, that molecule, that molecule, and that molecule to get to place B.
So one of the things that surprising in your book, at least I think will be to many readers, is that the number of neurons in the human brain, it actually peaks before birth and declines. And you write in the book so that by adulthood, we only have about half of the original neurons. What determines which ones survive?
Some neurons are born and do a transient job in making the brain. There's some great studies that are being done now that talk about how there's some very early born neurons that get connected up, and they help establish circuits. And later those neurons die because they've done their job they're like scaffolding of the brain. But most of the neurons that die are because they're competing with each other. So imagine players that want to join a football team. And the coach says, okay, we're going to form a new football team. It's going to have fantastic salaries. And there's a whole lot of players who want to play for this team. So they try out for the team. And half of them are cut because they're not as good as the other half. Well, there's no coach involved, of course, in making the neurons that populate the brain, but they compete with each other. Have I made a good synapse? Have I made a better synapse than you? Have I made a sufficient synapse to get enough feedback from the neuron that I'm connected with to say, you've done a good job, you should survive, but my neighbor here didn't do such a good job? That way you can assure that everything is connected up by the best ones that are effective. It seems to work kind of like that. Neurons compete for a limited amount of survival factor that's released by their target cells. And only those that get enough of it by making good synapses survive.
Yeah, the thing that surprised me about reading that was, I'm used to the whole idea of this from Gerald Edelman, but what really surprised me was the fact that it's mostly a chemical signal, not an electrical signal, that the electrical signal part doesn't seem to play a very big role, at least not in the developmental half of the equation.
Oh, no, I think that is wrong.
Okay, maybe I misunderstood.
Yeah, I think that the neurons competing, they are using the efficacy of the synapses that they're making, both the ones that they're making onto cells and the ones that they're getting from cells. The efficacy of those synapses is an important factor in determining whether they survive or not. Because if you block that all, or if you over-engage it, you can affect the survival of the cells. So if you block it all, then basically cells don't die because they don't outcompete each other. You need this differential competition, and that the differential competition is in a large part governed by the efficacy of the connections that are made. Not always, but largely.
So the presynaptic signal is the electrical signal, but that has to be strong in order to get the signal that goes to the postsynaptic neuron, the one that releases the trophic factors that's going to make it grow. Can't happen without a good electrical signal to begin with, is what you're saying.
And that's right. The presynaptic cell stimulates the postsynaptic cell. The postsynaptic cell says, thank you very much. Here's a little survival factor for you. That survival factor then is taken up by the presynaptic cell. It's rushed back to the nucleus and it says, survive, don't die, because the cells are ready to die if they don't get this.
Well, I wish that we could spend more time on that because it's really fascinating, but I think we're kind of running a little bit behind schedule. I apologize for not having better time management. Bill, what else would you like to share before we close?
I wrote this book to try to give a flavor of the field and give people an idea of what is going on inside an embryo's brain. Because every baby is born with its own brain. Like you're born with a particular face that might be a combination of your father's, your mother's, but it's partly your own too. Your brain is also your own. Because of all the complications, the thousands and thousands of steps and molecules and genes involved, no two people's brain is ever going to be built the same way. So it's just interesting for people to understand that we're not born just as pure, blank, equal slates. Everybody's different in their own way.
Do you have any advice for students that might be interested in going into this field?
Well, there's a lot of territory to cover still. We understand one cell of a Drosophila, how it gets made. We understand very little about a lot of how other cells get made. It's important medically, too, to make progress in brain diseases and brain repair. I think to understand how it was built in the first place. So there's motivation to do it medically, but there's also motivation to do it because it's a fascinating, difficult problem where there's lots and lots of territory yet to explore.
And I know you're retired, but what do you think is the most exciting unanswered question at this point in time?
I don't have one. I probably have lots, but I'm sorry, I don't have one that just leaps to mind as the most important one. And I don't want to go into one that may seem a little parochial, but so I'll leave that open. There's lots.
Fair enough. Fair enough. Well, I really appreciate you taking the time to talk with me. This is a really good book, and I don't feel like I've done a fair job of allowing you to share the key ideas, but I will definitely encourage my listeners to read this book because it really is a good book for listeners of all backgrounds.
Well, thank you very much. Maybe I talked too much and got you off track.
It's one of those topics, that is a little bit hard to visualize, right? And so when you're trying to put it into just audio, it's a challenge. And I think you did as good as you could with the questions I asked you.
Well, that was a challenge in writing the book, too, because textbooks are full of diagrams with thousands of labels. I wanted to be able to do it in words. So
in the book, you did a good job of limiting the diagrams to a manageable number, as being past the student stage. One of the things I find is when I try to read a textbook about something, all the diagrams become a little bit overwhelming. So it's nice when somebody you'd like to be able to read through, and if you want to look at the diagram, you can, but you don't have to. And you really did do a good job.
Cheers. Thank you so much.
Before I summarize the key ideas of today's interview, I want to remind you of a few items. First, don't forget to sign up for the free Brain Science newsletter. Just go to brainsciencepodcast.com or text Brain Science, all one word, to 55444. When you sign up, you'll get a free gift entitled 5 Things You Need to Know About Your Brain. That's Brain Science, all one word, to 55444. I also want to take a moment to thank those of you who support my work via My Lipson Premium subscriptions, Patreon, and direct donations. Brain Science is independently produced, so it relies on the support of listeners like you. You can learn more at brainsciencepodcast.com/donations. If you haven't already done so, I would appreciate you completing a short audience survey. There is a link for this at the top of this month's show notes. Finally, I am planning to take a cruise down the Rhine in April 2023. It will start in Amsterdam and end in Switzerland. I'm hoping to give a talk and have some listener meetups in Amsterdam and Zurich, and maybe just a meetup in Cologne. Please email me if you want to attend one of these events or if you want to help with the planning. I want to thank Dr. Harris again for coming on Brain Science to talk about his book, Zero to Birth, How the Human Brain is Built. Brain development is a complicated subject, and this book is a great introduction to how we know what we currently know. It's not a beginner-level book, but I recommend it to everyone working in other areas of neuroscience or medicine. Whether or not you choose to read this book, the goal of this review is to emphasize a few key ideas. Before I talk about brain development, I want to highlight a few other points. One of the big themes that comes from this book is the idea of evolutionary continuity. We have so much in common with other life, even single-celled organisms. As Harris pointed out, even paramecia generate action potentials or voltage spikes, even though they use calcium channels instead of sodium. They also use signaling proteins, the same ones that are neurotransmitters in our nervous system. I did mention Seth Grant because he was the first one who made me aware of this. We also have continuity with invertebrates like fruit flies, and our basic nervous system and our body plan is shared with all vertebrates. How does this happen? Well, every cell in our body contains the entire genome. So it's which genes are actually turned on that matter. Some genes code for transcription factors that turn on other genes. And so they can have a much bigger impact, especially if they turn on many other genes. So I want to start out by just reviewing the basic steps of brain formation. And I'm going to leave out most of the terminology to focus on the key ideas of what happens. We start out as a ball of pluripotent cells that develops 3 layers that are going to become the main tissues of the body. The outer layer is called the ectoderm, and it will become skin or nervous tissue. During a process called gastrulation, a section of the ectoderm, called the neural plate, gets pushed inside the embryo or pushed inside the ball and eventually forms the neural tube, which is what will become the brain and the spinal cord. This happens in all vertebrates. Once this basic plan has been established, the neural stem cells divide into a combination of stem cells and neurons, so that at birth we have most of the neurons we will ever have. The brain continues to grow after birth due to the growth of both connections and glial cells. Once the neurons have formed, they have to begin to make connections. This is a process that continues after birth and obviously throughout life. Finally, the neurons begin to send action potentials to each other and to tissues like muscles. It's kind of ironic that once the synapses are formed, the neurons actually have to compete for survival. I want to go through this again so that you can get it solid in your head. The pluripotent cells become part of the ectoderm and then the neuroectoderm, which is what will become the nervous system. During gastrulation, a structure called the neural plate is pushed inside the embryo and becomes the neural tube. The brain and spinal cord develop from the neural tube. Once this basic structure is in place, the neural stem cells begin to become neurons. The neurons send out axons to connect to other neurons or tissues such as muscles. Some neurons actually die before birth because they don't form successful synapses or because they have been part of a scaffold for building the brain and are no longer needed. We actually have the most neurons we will ever have before we are born. And at the time we are born, we will get very few additional neurons because neurons cannot divide or reproduce. Glial cells and synapses will continue to form. Obviously, brain development is extremely complex, and during our conversation, we barely touch on some of the fascinating discoveries about how it works. If you're interested in learning more, I highly recommend this book, Zero to Birth: How the Human Brain is Built by William A. Harris. We did explore a few key questions, like how does the basic body plan, including the brain and the spinal cord, come about? And since there are thousands of types of neurons, what determines the identity of any particular neuron? And how do the axons reach the right target? Harris mentioned just a few of the scientists who have worked on these questions going back to the early days of embryology, before we had the complex tools of molecular biology that now allow actual manipulation of genes to see what will happen. One of my favorite parts of this book is the descriptions of some of the groundbreaking experiments that have been done, some under extremely challenging conditions. Another cool thing is the surprises that have occurred along the way. For example, for years it was assumed that neural stem cells needed a particular signal to become neurons. But as Harris mentioned, it actually turns out that the ectodermal cells will become neurons unless they get a signal that tells them to become skin cells. Sort of like the way a fetus will be a female if it doesn't get the signals to make male sex organs. Another surprise we touched on during our conversation was the recent evidence that rather than evolving many times in the past, the key components of vision may have a common origin that goes back to the genes for photosensitive proteins in bacteria. Harris mentioned the experiment that showed that the human PAX6 gene can be inserted into a genetically blind fly and its compound eyes will form normally. That seems almost unbelievable given how different human eyes are from the compound eyes of flies. Even though this is a podcast about neuroscience, I think it is important to have a basic understanding of how the tools of molecular biology and genetics are integral, not just to neuroscience, but to every life science. What does it mean to say that we share much of our genetic makeup with other animals, like fruit flies, and single-celled organisms, like bacteria? The key is remembering what genes actually do. They code for making proteins, and proteins can have a huge variety of functions. Some become parts of complex structures, but others are transcription factors, which means that they turn other genes on or off. Some of these transcription factors seem to turn on whole cascades of other genes. This means that they might have similar but different functions in different animals. An example of this is the Hox family of genes. That's spelled Hox. We talked a little bit about the Hox family of genes, which are arranged in a sequence that corresponds to the head-to-toe structures of the body. They were first discovered in fruit flies, and a scientist named Ed Harris showed that if the Hox genes were inserted into other cells, it was possible to grow extra wings or extra legs where they wouldn't normally be located. However, Bill Harris emphasized that these Hox genes don't code for chemical signals. They actually code for proteins that are transcription factors that may turn on thousands of other genes. Unless you work in this field, it's not necessary to worry about the details. I picked the Hox gene as an example because it is one of the ones that is found in invertebrates like fruit flies. But they only have one set of 8 Hox genes, while vertebrates have 4 sets. This is because the sets were apparently duplicated twice during early vertebrate evolution. I first learned about this gene duplication, which I think occurred about 250 million years ago, from Seth Grant. And I think it is a very underappreciated element of why vertebrates were able to evolve into more complex and diverse species. I want to close by mentioning a point that Harris emphasized near the end of the interview. At birth, each baby's brain is unique. It's not a blank slate. I would love to hear your feedback about this episode or anything else. You can write to me at brainsciencepodcast@gmail.com, and you can find complete show notes and episode transcripts at brainsciencepodcast.com. It's been many years since we talked about meditation, and next month we will explore this topic with Evan Thompson. Many of you may remember him from our discussion about embodied cognition, but Thompson is also a philosopher with an extensive knowledge of Eastern thought. His background gives him a unique perspective, and we will be critically examining the claims that meditation is a mind science. Until then, I hope you will check out my other podcasts, Graying Rainbows and Books and Ideas. Thanks again for listening. I look forward to talking with you again next month. Brain Science is copyrighted by Virginia Campbell, MD. You may share the show with others, but for any other uses or derivatives, please write to brainsciencepodcast@gmail.com for permission. The theme music for Brain Science is Mind Fire, written and performed by Tony Catraccia. You can find his work at syncopationnow.com.